Photosynthesis is a process by which plants and photosynthetic bacteria use sunlight, water, and carbon dioxide to produce a simple sugar, glucose. These plants and photosynthetic bacteria photosynthesize to make glucose. These organisms then use the glucose as food. This food is used as energy, which allows photosynthetic organisms to perform work.
There are two stages of photosynthesis. One is called a light-dependent reaction, while the other is called the Calvin cycle, or the light-independent reaction. The first stage, the light-dependent reaction, requires light and water to create ATP, a vessel of chemical energy, oxygen and NADPH, which serves as the electron carrier, also defined as the charged battery of the process. This takes place in the thylakoid membrane, which is an outer layer surrounding a thylakoid. Inside the membrane, chlorophyll absorbs light energy and excites electrons, which generates energy.
The remaining electron is caught
by NADP+. This becomes NADPH. Thylakoids, when stacked on top of one another, form a granum structure. This granum structure is found inside the chloroplast of a plant cell. Chloroplasts contain a colorless fluid called stroma that surrounds the grana structures. This is where the second stage of photosynthesis, called the Calvin cycle, light-independent reaction, takes place. In this stage, chemical energy, ATP, is used to convert CO2 into glucose. When NADPH loses an electron, it turns back into NADP+.
The use of bipyridinium as a pesticide was permitted in 2004 by the European Union. There are two forms known as diquat and paraquat. Diquat is an aquatic herbicide and is used with crops like alfalfa, clover, potato, grain, sorghum, and soybean. It is used to spray trees and crops for weed control. Paraquat, on the other hand, is used in pre-harvest and has a desiccant use. It is a soluble concentration and is used quickly kill vegetation as needed to plant another crop.
Paraquat isn't used by homeowners because it is classified as restrictive use. Diquat is a homeowner product.
This inhibits photosynthesis because, when exposed to sunlight, bipyridinium herbicides form oxygen compounds like hydrogen peroxide. These oxygen compounds regenerate forms of oxidized paraquat. In light reactions, this paraquat destroys electrons in photosystem 1 that would have otherwise made NAD+ into NADPH. Without NADPH, the Calvin cycle cannot take place, therefore glucose cannot be produced.
Cellular respiration is the act of making ATP from glucose through the mitochondria to make energy for the cells to do their work. The balanced equation is the reverse of the photosynthesis equation. This process is a catabolic reaction, which means its reactants break down to create something smaller. The reactants are glucose and 6 molecules of oxygen. The products are 6 molecules of carbon dioxide and 6 molecules of water. Glycolysis is the first stage of cellular respiration, in which glucose is first being broken down into 2 molecules of pyruvate. Occurring inside the cytosol, also called cytoplasm, ATP is processed to make 2 more ATP, 4 ATP. First, to break the glucose bond, ATP exerts itself into the bond, which releases electrons. Those electrons combine with NAD+ to create NADH, which serves as an electron carrier, or a charged battery. The releasing of electrons also creates 4 ADP, which becomes 4 ATP. This is when the glucose bond splits into 2 pyruvate.
The Krebs cycle, also called the citric acid cycle, is the second stage. This stage takes those 2 pyruvates created from a single glucose bond and breaks them into FADH2 and 3 NADH, completing the breakdown of glucose. All this occurs inside the mitochondrial matrix, a space inside mitochondria. In the cycle, the 2 pyruvate make 2 molecules called acetyl-CoA, which produces 3 molecules of carbon dioxide. When those bonds are broken, 3 molecules of NAD+ create 3 molecules of NADH, and FAD+ reacts with the cycle to create FADH2. Finally, ADP creates 1 molecule of ATP.
The third and final stage of cellular respiration is called oxidative phosphorylation, which generates ATP through the electron transport chain. This process is located in the intermembrane space, which is right in between the inner and outer membrane of the mitochondria. The electron transport chain consists of several protein complexes in an ATP synthase. In oxidative phosphorylation, NADH loses an electron and becomes NAD+. The electron is passed in between protein complexes until it reaches the end. When it reaches the end, this electron reacts with an oxygen and hydrogen molecule and creates a byproduct of water, H2O. Hydrogen molecules move through the protein complexes into the intermembrane space. When this space becomes too concentrated, these hydrogen molecules move through the ATP synthase into the mitochondrial matrix. The movement of hydrogen through the ATP synthase creates kinetic energy, which is converted into chemical energy by ADP to ATP.
The metabolic inhibitor is known as oligomycin. Oligomycin are macrolides created by streptomyces that can be poisonous to other organisms. Some symptoms are nausea, skin rashes, eye irritation, and inhalation causes respiratory tract irritation. It's caused by prolonged heating of oligomycin and is used as an antibiotic. Some treatment is using gloves and goggles when handling the metabolic inhibitor. Inhibits cellular respiration by stopping the oxidative phosphorylation step by blocking the proton channel of the ATP synthase. This results in the reduction of electron flow. Without the flow of electrons, energy cannot be produced, resulting in less conversion of ATP to ATP.